Welcome back, everyone, to the Connect Science webinar. It's a tremendous honor today to have Dr. Tyler Jacks speaking on dissecting of tumor evolution at single-cell resolution. Tyler is well known to the community. He's a professor of biology at MIT, a Howard Hughes Medical Institute investigator, and director of the Koch Institute for Integrative Cancer Research at MIT. Not only has he made a series of seminal discoveries in cancer biology, but he's trained many of the leading cancer biologists around the country. Before we start, as always, I invite you to post your questions using the Q&A tab, and Dr. Jacks will, time permitting, address as many as possible. I also thank the Leukemia Lymphoma Society and the Dana Farber Harvard Cancer Center for supporting this webinar. And at that point, Tyler, please take it away.
Great, David. Thank you for the introduction, and thanks to you and Nellie for the invitation. It's a pleasure to join all of you in this fashion. It's noon in Newton, Mass. I don't know what time it is where you are. I'm going to be describing our work today related to understanding tumor progression or tumor evolution in the context of models of lung cancer and then pancreas cancer that we've developed in my laboratory over this last several years. I have connections to many companies as listed here, but none of those connections influence any of the conclusions or interpretations of data that I'll describe for you today. By way of general introduction, we know that cancer cells arise from normal cells through the acquisition of mutations in oncogenes and tumor suppressor genes. And these mutations are clearly very important in determining the features and phenotypes of cancer cells. But we also know that other changes, epigenetic changes, non-genetic changes, are also important, very important, in determining the various emerging properties of cancer cells that allow them to successfully navigate the various stages from normalcy through malignancy. In addition, we know that cancers do not arise in isolation, but in relation to other cells in the body. And these other cell types can both positively and negatively affect the tumorigenic process. So in today's seminar, I'm going to divide it into two parts. The first part will last about 2/3 of the seminar and relates to two recent stories that revolve around this point of epigenetic or non-genetic factors that regulate tumor progression. And this was done at the single cell level, and the work that I'll describe is all unpublished. In the latter portion, I'll discuss recent work, again unpublished, that involves our work related to interrogating the role of the adaptive immune system, and particularly T cells, in controlling tumor progression in a model, or a few models actually, of pancreas cancer. So to get started, most of the projects in my lab involve genetically engineered mouse models of cancer, and the one that we use most extensively is a model of lung adenocarcinoma. We call it the KP model. It involves the conditional activation of an oncogenic allele of KRAS, made first in my lab by David Tubison. And in this particular version of the model, we've also combined in a floxed allele of P53 that was made by Jos Jonkers and Anton Burns's lab, thus the K and the P. And these two mutations are quite common in human lung adenocarcinoma as well. We activate RAS and inactivate P53 by exposing the animals to a virus that carries Cre recombinase. RAS skin gets activated, P53 deleted in individual cells of the lung, and that's enough to initiate tumor development in situ. These tumors will progress locally, and then at some frequency will metastasize. And about 30% of this or so of the mice at end stage, and that's about 6 months or so in the KP model, will have lymph node and distant metastases. Now, we can evaluate the progression of the disease in many different ways. For many studies, including work from Carla Kim in my lab, but many other investigators, we believe that in this model, as well as in human lung adenocarcinoma, a major cell of origin of the disease is the alveolar type 2 cell, or AT2 cell. In the early stages of disease development in this model, within a few weeks, we observe adenomatous hyperplasia, as illustrated here, followed by adenomas, benign lesions, Within about 4 months or so, we see a transition to real cancer, adenocarcinomas. These will become locally invasive in time, and as I said, eventually a fraction of them will metastasize. So one of the values of this model and models like it is that we can interrogate the various stages of tumor development and try to understand what the arrows at the bottom of the slide represent in molecular terms. What are the drivers of these transitions? And that's illustrated here. And this has been a focus for our work, I would say, for probably 20 years, actually, since we started working on this model. Now, you might imagine in the current era of cancer genomics that these transitions would translate into additional acquired somatic mutations. That KRAS and P53 are enough to get started, but you need to acquire additional mutations to drive the various subsequent transitions. That's what we thought would be true until David McFadden, working with Katie Politti and Greg Hannon and Harold Varmus, sequenced many of these tumors, including several advanced tumors, and found to our surprise that they had very few acquired mutations, very few somatic mutations. And on average, these tumors had about 2 to 5 protein altering mutations per tumor, and there were virtually no recurrent mutations. And so, therefore, one has to conclude that the transitions from early stage to late stage are not driven by genetic alterations, at least as detectable by genome resequencing. I should say that there are some genomic alterations at the copy number level, including the gain of KRAS itself, the oncogenic allele, as well as other reproducible changes in copy number and other chromosomes. But by and large, these genomes are relatively quiet. And therefore, we came to the conclusion that these transitions were likely not driven by genetic alterations, but rather by epigenetic alterations. And we'd actually acquired a lot of information over the years that supported this view. Monty Winslow, when he was in the lab, showed that the transcription factor NKX2.1, which is an important lineage-restricted transcription factor in the lung, went from being highly expressed in early stage tumors to lowly expressed or not expressed at all in late stage tumors, as shown here. This is not a genetic event. NKX2.1 becomes epigenetically silenced in the development of this disease. Other markers became upregulated during tumor development, like HMG-A2 here, which is normally expressed in embryogenesis, but becomes ectopically expressed in advanced tumors in this model, as well as other mouse models, as well as in human cancers. So again, this represents some sort of gene expression alteration that may represent a loss of differentiation state and a gain of a more embryonic or stem-like state. In addition, Eric Snyder, when he was in the lab, demonstrated that upon loss of NKX2.1, tumors developed an interesting and surprising gastric-like phenotype marked by the expression of a transcription factor called HNF4 alpha, which is normally expressed in the intestine and in the liver. And that was surprising. Why would lung cancers adopt this more gastric-like phenotype? We think it probably has something to do with the fact that in embryogenesis, the lung actually develops from the gut. And therefore, it may be that tumor development is somewhat of a representation of normal development gone wrong or gone backwards in this case. So with that sort of background, we decided to explore these questions at greater resolution. And in the current era of single cell resolution, we thought we were particularly well positioned to do so. And I should say that the work I'm about to describe was a large collaboration, very importantly involving Aviv Regev's lab and Matan Hoffrey and her group and others. And it was started in my lab by Nemo Marjanovic, who's a joint student between me and Aviv, and Tuomas Tamala. Tuomas now has his own lab at Memorial Sloan Kettering, and he has carried on this work, and his group has contributed important recent information. So, as I've mentioned, this model develops along very well-defined histological stages, and as such, we can collect cells from these various stages and profile them by single-cell RNA sequencing. When we began this work, 10x sequencing was in its infancy, and so we used SMART-seq2 instead, where one sorts individual cells and sequences them directly. that method has the advantage that we get a greater depth of sequencing per cell, about 4,000 to 5,000 genes scored per cell, although the throughput is lower. So in this study, we've sequenced about 4,000, slightly more than 4,000 cells in total. And again, these cells are isolated from different stages: early lesions, middle stage lesions, and late stage lesions, and multiple of them in each case. The early lesions are a combination of KRAS-only mutant cells and KRAS P53 mutant cells, and the latest stage lesions represent KP at the advanced stage. So sequenced the 4,000+ cells and then began to analyze them, initially using t-SNE analysis, which many of you will know is an algorithm that allows highly multi-dimensional data to be reduced to a two-dimensional representation, where each dot on these plots represents an individual cell, and its position is an indication of its place in transcriptional space. And two cells that are close together on such plots have more similar transcriptional programs, transcriptional states, let's say. Now, I've grayed out the identity of the cells in this representation, so it's a little bit hard to make sense of it. Makes a little more sense when I show you where these cells came from. And here I'm using the color coding that I've indicated at the top. And I should have mentioned that all of these samples have a pre-activatable TD tomato allele, and so they're easily sorted away from all the other cells in the tumor or tissue. So we're only looking at the cancer cells, or in the case of the AT2 cells, the normal counterpart. And the normal cells are colored in green, the early lesions in shades of blue, and then the middle lesions in orange, and the late lesions in red. And so you can see that we have a pattern emerging here from normalcy to more advanced malignancy in a direction that seems to follow a path that looks like this. So this is a static picture. I actually think it looks more interesting if you, in a sense, show the movie of tumor progression. So I'm gonna let the movie run in a moment, and you'll see that whereas we start with a nice tight cluster of alveolar type 2 cells that have a similar program of gene expression, as we begin to move through tumor progression from early stage to later stages, the cells begin to move away from that normal pattern and also become more heterogeneous in time. So here are the earlier stage lesions, and then the middle stage lesions, and then the later stage lesions. And again, I think you can see two important features here. One, as tumor progression proceeds, the cells become less and less similar to the cell of origin. And in addition, as tumor progression proceeds, we see a greater degree of heterogeneity emerging. Now, the heterogeneity is interesting, but it also could be trivial. These late stages represent multiple distinct late stage tumors. And you could argue that these clusters of cells at the late stage, which you can imagine exist here, 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 and here, simply represent different tumors at the late stage. And you can see them in isolation here. Here are just the 30-week time point KP cells derived from multiple grade 3 and grade 4 carcinomas, individually isolated from animals. And again, you could argue maybe this is one tumor and that's a different tumor and that's a different tumor, and they simply chose different paths to get to this late stage. But that's actually not true. We kept track of which tumor gave rise to which cells, and if we map the identity of those tumors shown here on these various clusters, you'll see that the major clusters actually have representation from most of the tumors that were profiled. And that is a very interesting and frankly surprising observation, that there is a fairly well described, not quite stereotyped, but well articulated path that cancer cells take as they move from the earlier to later stages, a more constrained path towards late stage tumorigenesis than you might have imagined, which we ascribe to the sort of developmental history that the cells have had, as well as the epigenetic state that they started from. There's obviously an influence of the mutations that we've programmed in, but this is not a chaotic process. There seems to be a more limited set of roads to roam. Okay, so now let's dig a little bit deeper into these data and see what we can learn. Firstly, with respect to information I've already told you about, I mentioned that we had shown that NKX 2.1, which is a transcription factor that controls the expression of many of the important genes involved in alveolar type 2 cells, was downregulated during tumorigenesis. And indeed, you can see here that surfactant protein C, which is one of the targets of NKX2.1, is highly expressed, indicated by this deep red color, highly expressed in the cells of origin, as well as the early stage lesions, but its expression is lost over time. Very consistent with that image I showed you before of NKX2.1 levels in early versus late stage tumors. We also talked about that gastric phenotype that emerges as tumors develop. I told you about a marker, HNF4 alpha, and you can see here that HNF4 alpha is not expressed in the cells of origin or in the early stage lesions, but it becomes expressed as cells transition through these middle stages of tumorigenesis. And finally, here's HMGA2, which is expressed in an even more restricted set of cells associated with the latest stages of tumorigenesis. These tumors do show features of the epithelial mesenchymal transition, which is associated with progression towards metastasis, and a small subset of cells in this cluster here show that EMT marker ZEB2, and this likely is a cluster associated with the very latest stage of progression. And we'll return to that subject later in the presentation. So I showed you the description of, or representation of the data by t-SNE plotting. There are many ways to do dimension reduction now with single cell data. Fate mapping is another, and this is a version of the same data now illustrated by fate mapping. And you can see that the basic contours are similar and patterns are similar to what I showed you with the t-SNE. The color scheme is the same. Fate mapping is preferred by some because the local architecture is better preserved in fate maps than it is in t-SNE maps, and so you can draw more conclusions about clusters of cells that are nearby to one another by this sort of mapping. And Matan Hafri, who did all this computational analysis, used fate mapping to begin to explore the patterns of gene expression that might be associated with these various groupings of cells. And he made the observation, which we had not made before, that in this zone here, there was a clear transition from pure alveolar type 2 phenotype or gene expression pattern to one that represented a mix of both alveolar type 2 and alveolar type 1, another major cell type within the alveolus. And we had not appreciated that previously from our earlier characterization of these tumors. You can see it quite clearly here. And this is a marker of type 1 cells, HOPX, and you can see it's not expressed in the cells of origin or the early lesions, but it becomes expressed as cells transition towards the middle stages of tumorigenesis. It's actually known that there's a close relationship between type 2 and type 1 cells, both in embryogenesis, where they arise from a common progenitor. Also in the adult lung, Carla Kim in my lab showed that there was at least bipotent progenitor, maybe stem cell. She described it as BASC, which can give rise to both of these cell types. And other workers have shown that type 2 cells actually can be a source of type 1 cells, including an injury repair. And Mark Krasnow and Tushar Desai have shown very nicely that there's a subset of type 2 cells which have a stem cell-like phenotype, and under homeostatic and injury conditions can give rise to type 1 cells. So there's a close connection between type 2 and type 1. And we think what we're observing here is that the cancer cells are sort of representing an injury response where the cells are beginning to elicit this program that normal type 2 cells will do under stress and injury repair conditions. Here's the fate map once again, and I said that it can be used to cluster cells in different groupings, and one can give those particular clusters numbers, as was shown here. In this particular analysis, we described 12 clusters of cells that are grouped together based on their similarities in gene expression programs. By doing this type of analysis, we began to focus our attention on cluster 5, this blue cluster towards the top, and I'll explain why it became so interesting to us in a moment. Cluster 5 shown here can be marked by a couple of specific genes, SLC4A11, which you'll see we use in immunohistochemistry analysis. This is a solute borate co-transporter. Not clear that it's interesting with respect to cancer biology, but it serves as a useful tool to interrogate these cells in histological samples. And here's TIGIT, which you may know most as an inhibitory receptor in T cells and other immune cells. Again, not clear why it's expressed in this population of cells, could be biologically interesting. But for us, it serves as a marker for purifying these cells, as you'll see in a moment. So cluster 5 became interesting to us because despite what I said before, that the cells march through from early stage to late stage in a progression that goes roughly from south to north, it turns out that cluster 5 is quite well represented, not just in the late stage cells, late stage tumors, I should say, as indicated here. The percentage of cluster 5 cells with a cluster 5 transcriptional profile is fairly consistent across all these late stage lung adenocarcinomas. But note that cluster 5 cells exist in early lung adenocarcinomas and even in the adenomas, the more 12-week time point cells that have not yet made the transition from a benign to a malignant tumor. The presence of these cells at early stages and the persistence of these cells at fairly defined frequencies was quite interesting to us and could be interpreted many different ways. It could be that these cells have certain stem-like qualities and that they reproduce themselves starting from this point forward and therefore maintain their numbers in roughly consistent levels. Alternatively, it might be that the presence of these cells represents a flow of cells from other transcriptional states into the cluster 5 state and then out from the cluster 5 state. That is to say, the cluster 5 is being regenerated and diminished sort of consistently over time. And that seemed interesting, and I'll say more about it in a moment. Using this SLC4A11 marker, we could show in situ that grade 4 tumors in the KP model, grade 3 tumors showed scattered positive cells, and even grade 2 tumors would show some scattered positivity. Also interestingly, when we took the gene expression signature from cluster 5 from the mouse and used the homologous genes from human and analyzed single cell RNA sequencing data from a small subset of samples from 2 different papers, we observed that cluster 5 cells were present in these samples as well, interestingly at similar proportions as we observed in the mouse. And when we use SLC4A11 on tissue samples from human tumors, once again, we can see scattered positivity. So the cluster 5 cells, the cluster 5 phenotype seemed interesting for all of those reasons. As I mentioned, one possibility was that the existence of cluster 5 cells in this sort of conserved proportion could be a function of the fact that cells move into this cluster and move out of this cluster, making cluster 5 having a high degree of plasticity. And to analyze that, Matan used Waddington optimal transport, which is a way to evaluate from a collection of single cells where one marks a beginning point and an ending point. Here it's the AT2 cells as the beginning point and the cluster 11 cells, these cells, as the ending point. The various routes that cells can take to get from one state to another. You can think of this as a sort of transportation map that cells can transition from state to state, and it's based on various computational parameters. And in this analysis, cluster 5 actually looks like the cluster for which many different states move into and many different states move out of. Again, suggesting that this is a highly plastic culture that has perhaps, again, more stem cell-like features. I find these kinds of maps a little confusing to look at. So we've broken it down here and just stripped away some of the unnecessary information. If you look at cluster 11, which was one of those late stage clusters here, no clusters emerge from that cluster. We only see paths towards that cluster as indicated in the purple color. Whereas cluster 5 is much more interesting. Several clusters give rise to cluster 5 as shown here, and cluster 5 gives rise to many other clusters, at least as predicted by this algorithm. So again, the suggestion is that cluster 5 has high plasticity, that it's a high plasticity cell state. Now, if it were a high plasticity cell state, the prediction would be that those cells would have certain interesting features with respect to tumorigenesis. And Thomas and his group have explored that in different ways. For example, they've isolated or purified or enriched the cluster 5 cells using TIGIT as a marker. They've taken tumors and isolated the TIGIT positive cells, the TIGIT negative cells, as well as cells from cluster 11, which is marked by CD109, and then placed those cells in 3-dimensional culture and allowed them to grow for a period of time. If the cluster 5 or high plasticity state cells have a greater opportunity or ability to give rise to cells with distinct transcriptional states, one might be able to read that out as they propagate in culture, and that may be less true of the cells which are depleted of those cells or of the cluster 5 more fixed cluster. And that turns out to be true. If you then do single cell analysis at this late time point, you can see that the cluster 5 cells, the high plasticity state cells, have a greater range of transcriptional profiles as demonstrated here. The fixed state, the cluster 11 cells, don't do much interesting in the transcriptional space, and the depleted cells are somewhere in the middle. You can actually quantify the ability of cells to take on more heterogeneous transcriptional profiles using this type of analysis. And you can see that the cluster 11 cells are least capable of developing heterogeneity. They're more homogeneous. And the high plasticity state cells, cluster 5 cells, have the greatest degree. In addition, one might expect that the cluster 5 cells would have increased tumorigenic features. And Thomas's group has demonstrated that as well. If you plate these cells in 3-dimensional culture and count the number of tumor spheres per 500 cells plated, the TIGIT positive cells are much more active, much more able to form tumor spheres. And likewise, in a transplantation tumorigenesis assay, the TIGIT positive cells have increased tumorigenic potential. So in these ways, importantly, this high plasticity cell state seems to be associated with increased tumorigenesis, and therefore reveals an important feature of tumor progression. Finally, we've looked at the relationship between this program, illustrated by this high plasticity cell state, as it relates to human TCGA RNA-seq data. If this is a highly malignant state, then patients who have a high percentage of cells that show this same phenotype would be predicted to have worse survival. And that's indeed what we see. Looking at the various clusters and stratifying them based on highest to worst, best to worst survival, indeed the cluster 5, the samples that are enriched most strongly in the cluster 5 signature have the worst prognosis, again making an important association with the human disease. So I'm going to make a transition now and talk about a related but distinct data set motivated by this observation that tumor cells in this model progress along particular pathways defined by their gene expression programs. We, of course, wondered what was behind these transitions. What were the factors, the epigenetic regulators, transcription factors that controlled these transitions? What gives rise to the patterns that we observe? And to do this work, Lindsay Lefebvre, a postdoc in my lab, initiated a collaboration with Jason Buonrostro's lab. Jason is a professor in the Department of Cell and Regenerative Biology at Harvard. And Jason, as a graduate student with Will Greenleaf and Howard Chang, helped to invent attack sequencing. And so he's got great expertise in that realm, and members of his group likewise have great computational skills to interpret these very complex data. So we worked with Jason to apply attack sequencing to the same model, try to understand the epigenetic regulation of tumor progression. Attack sequencing was initially developed to explore the state of chromatin, open and closed chromatin. Open chromatin is more accessible by transposon insertion. These transposons are linked with sequencing primer binding sites that allow one to amplify the regions of transposon insertion. and thereby characterize the regions of open chromatin within a sample. And it was originally developed using bulk samples like bulk tumor or bulk normal tissue. But the information, as we've just been discussing, becomes much richer and much more interesting if one can look at the single cell level. And investigators like Jay Shanduri and others have developed single cell methodologies, and now there are quite a few of them. We use a modification of this one called SkyAttack, which is a combinatorial barcoding scheme for isolating the patterns associated with individual cells. And this works extremely well. This is an illustration of that. Here are data from a bulk sample, tumor sample, and you can see a series of peaks associated with transposon insertion. And here's a collection of single cell analyses from the same tumor, and you can see that the data look virtually identical. So the single cell data is actually quite reliable, reproducible, and robust. We've been extremely pleased. So what did we do? Well, analogous to the single cell RNA sequencing story, we looked at samples from the KP model, also marked by tomato, so we could isolate the cancer cells away from the normal cells. In this case, we did a variety of things. We took late stage KPT, tomato, tumor-bearing lungs. So these are animals that have been harboring tumors for some time. I'll show you that there's quite a bit of heterogeneity in these samples in a moment. We also isolated a series of late-stage tumors, as well as, not shown here, some early lesions. And we isolated metastases, lymph node, as well as some distant metastases. The late-stage tumor-bearing lungs actually themselves have quite heterogeneous patterns. Here's an animal with that was initiated 35 weeks prior. It has a bunch of different tumors. These have been graded actually using an artificial intelligence-driven algorithm for histology grading made by a company called Aphoria in collaboration with Peter Westcott and Thomas Tamala in my group. And this allows us to see quite quickly that these animals have quite a great deal of intertumoral heterogeneity with respect to stage. You can see a lot of grade 3 blue tumors, but also a lot of grade 2 tumors, and even some grade 4 regions as well. There's also intratumoral heterogeneity. You can appreciate here. Here's a grade 3 tumor with a patch of grade 4, a grade 3 tumor with a patch of grade 2. The point of this being that when you profile at 35.5 weeks, you're actually profiling cancer cells that have progressed different stages of tumorigenesis. So we looked using single cell attack sequencing in a very large number of cells. Lindsay and her colleagues profiled more than 13,000 cells by this method, 13,000 primary tumor cells, 600 cells derived from metastases, and these metastases were from lymph node, chest wall, as well as liver. She also found it useful to profile normal cells of the lung to give certain landmarks, and you'll see why that's important in a second. And I'm showing you some of those cells now, though not all of them. So a large number of cells went into this analysis, and those are mapped using another dimension reduction methodology called UMAP projection. And the cancer cells from the primary tumors are shaded in gray, the metastases, cells derived from metastases, in red, and normal cells, some of the normal cells are shown in blue. The data you'll see here looks more continuous than some of the data I showed you previously for the single cell RNA sequencing. And we think that's a function of the fact that there's actually many more data points. You're looking at many thousands of peaks in characterizing the epigenetic profile of these cells. But the same principles hold. These dots represent individual cells, and their position on this plot is a representation similarity to their neighbor here in the chromatin profiling space. Now, as I mentioned earlier, with respect to the single cell RNA sequencing profiling, you could say that there's a great degree of heterogeneity that exists across these samples. That could be a representation of simply different samples giving different profiles. But here again, we know that there's tremendous intratumoral heterogeneity as it relates to the epigenetic profiles because, as I mentioned, we've profiled some individual tumors. And when we look at an individual tumor or two, as shown over here, you can see that the cells represent different points in this progression, as illustrated here. So intratumoral heterogeneity is quite common as we evaluate it at this level. Now, we think that tumor progression proceeds from the left of this pattern to the right. And I say that for a number of reasons. Firstly, as I've mentioned, we know that the cell of origin for this disease is an alveolar type 2 cell. And we profiled alveolar type 2 cells, and the normal alveolar type 2 cells sit right here. And certain cancer cells sit very near to them. So we would imagine that these are cells that have an early stage chromatin profile or phenotype. We also had separately isolated metastases, and all of the metastases, interestingly, sit in a particular grouping shown here. And so crudely, you could say that tumors start from this side and move progressively towards that side. We actually have more evidence that that's probably right. Lindsay profiled some tumors from animals that were initiated only 8 weeks prior, so early stage lesions, and this is what they look like. You can see that there's some grade 1 cells and some grade 2 cells, and when you profile those by single cell attack sequencing, they all line up here, most of them overlapping with the alveolar type 2 cells. So it's safe to say that the cells start here and move in this direction towards the more advanced phenotypes, and we'll talk more about that momentarily. I want to highlight an interesting observation that Lindsay made with respect to this metastasis or metastasis-like cluster. You can see that the cells that are present within this grouping are mostly composed of metastasis-derived cells in red. But if you look carefully, you can see that there are quite a few primary tumor cells intermixed. And the suggestion from that was that primary tumor cells can adopt an epigenetic profile that looks very much like an eventual metastasis, leading to the possibility, at least, that as tumor progression proceeds, cells adopt a metastasis or pre-metastasis, pre-metastatic cell state, which enables them to become metastatic and ultimately seed and successfully seed a distant metastasis. Indeed, when Lindsay looked for cells in primary tumors that had this profile, obviously all the metastases, all the cells in metastases had these profiles. That's indicated by this cluster. But you can see that several of the primary tumors had cells that had this profile as well. And we again believe that these are cells that are on their way to becoming metastatic, pre-metastatic cells. We can't exclude the possibility that some frequency cells cycle back in a reseeding type phenomenon from a metastasis back into a primary tumor. But for a variety of reasons, we believe that most of these cells are actually on their way to becoming metastases. So we can look at these data in a fashion similar to what I talked to you about previously with respect to the single cell gene expression. By looking at the patterns of chromatin accessibility of AT1 cells and AT2 cells, we can overlay those patterns on our data. And indeed, we can find that some of our cancer cells have a pattern that looks just like AT2. Others, consistent with our earlier analysis, have a pattern that looks more like AT1. We can see this reinforced in immunofluorescence of early stage primary tumors. Here's a tumor that's stained for SPC, that marker of alveolar type 2 cells. Most of the cells in this tumor are SPC positive, but you'll see this little patch of cells which have begun to downregulate SPC, almost certainly through a downregulation of NKX 2.1. And here, these cells have begun to upregulate that marker HOPX, a marker of alveolar type 1 cells. That's that transition I was telling you about before. This is an example of lineage infidelity, lineage switching in the context of tumorigenesis. Taking the patterns of chromatin accessibility and overlaying that with transcription factors that bind to sequences within those accessible peaks, transcription factors and other regulators. Jason and his group were able to develop algorithms that allow the description of these data in various modules, various discrete modules. Module 11 has an AT2-like profile, and it does mark the cells that look most like the alveolar type 2 cells. Module 6 looks like alveolar type 1 cells, and again, overlays nicely with what we saw previously as the cells transition from AT2 towards AT1. Module 7 looks like a gastric phenotype, and I've mentioned that previously. That's that switch to a more gastric gene expression program that we've seen now a couple of times. Module 9 is associated with a transition from the earlier stage to the late stage, and I'll have more to say about that later. And Module 4 was most associated with that metastatic and pre-metastatic cell cluster, the most advanced cells. Module 11 and 6 are associated with NKX 2.1 accessibility. The NKX 2.1 motif is highly accessible within cells within these modules. And you can see that in specific examples here. Here's that gene SPC. It has two NKX 2.1 binding sites. And you can see that they're well accessible in early stage cells from early stage tumors. But in the late stage, those peaks are no longer accessible. NKX 2.1 has been downregulated. This gene has been shut off. HNF4 alpha is one of the genes that drives this module, and you can see that it's highly accessible in the cells that show this particular pattern, this particular profile or phenotype. Module 2 is associated with a transcription factor called NKX 2.1, which overlaps in part with this module 9, this late stage module. And this transition towards the late stage was very interesting to us. One of our goals in carrying out this analysis was to understand the transition from early stage to late stage. And RUNX2, a transcription factor that's not normally associated with lung biology, most associated actually with bone development, becomes highly accessible in the late stages. This is an example of RUNX2 gene expression, RUNX2 gene activity score by chromatin profiling. There's low accessibility in the near the sites of initiation of RUNX2 in early stage lesions or cells from early stage lesions, but at the late stages, one can see accessibility. RUNX2's gene score goes up in the late stages, and its motif becomes accessible in the late stages. We had no prior understanding of RUNX2 biology in lung tumor development. It has been associated with other cancers, in breast cancer, certain leukemias as well, but had not been closely associated with lung tumor development. But this clearly showed a potential role for RUNX2 in controlling this transition from the early stage to the late stage. We've explored RUNX2 more extensively. We can show by immunohistochemistry that RUNX2 is expressed at high levels in individual cells within later stage lesions, as well as in metastases, and in a subset of human cancers. Here from the Human Protein Atlas, human lung adenocarcinomas show spotty expression of RUNX2 as well. In order to probe the biology of RUNX2, we've carried out CRISPR-based modulation of RUNX2 function in cell lines derived from the KP tumor. Cell lines that are known to be metastatic, which express high levels of RUNX2, we can use these to inactivate RUNX2 by CRISPR inactivation. In cell lines that are earlier stage, non-metastatic, express lower levels of RUNX2, we can increase the level of RUNX2 using CRISPR-A. And in those ways, we can ask, does the chromatin profile of those cells change in a fashion that's consistent with the data from the tumor profiling? And we can also look at the biology of these cells by functional analysis. Does RUNX2 control genes and programs that you would associate with progression to malignancy, progression towards metastasis? Do the cells that have modulated RUNX2 function change in those properties? With respect to the first point, when we compare RUNX2 knockout cells with RUNX2 overexpression cells and look at the differential accessibility of various genes, we can characterize them from most different to least different. And to our interest, many of the genes that were found to be increased in their accessibility as a function of RUNX2 status were genes that encoded proteins associated with the extracellular matrix and the matrosome, as though RUNX2 was controlling a program that allowed these cells to contribute differentially to the extracellular matrix, become more in the EMT transition, more towards the mesenchymal state, properties that we know are important in tumor progression towards metastasis. We can actually see that very clearly in immunofluorescence. The most differentially expressed gene is here, galectin-1, a lectin. You can see that this gene is expressed in regions of tumors like that shown here. This is a tumor in transition. This is a very late stage region. It's associated with upregulation of RUNX2 and upregulation of galectin-1 and downregulation of NKX2.1. This is this transition to malignancy that we're talking about. We would imagine that this is one of those or a set of those pre-metastatic cells that are on their way to becoming metastases. And indeed, galectin-1 gene score, activity of galectin-1 is predicted to be very high in those metastatic cells and pre-metastatic cells present within this cluster. And finally, or nearly finally, we've also addressed functionally the importance of RUNX2 in metastasis by taking those cells that were known to have metastatic potential and express RUNX2, inactivating RUNX2 by CRISPR inactivation. We could then test their metastatic capability using tail vein assays. We inject those cells in the tail vein. The control cells will make many tumors in the lungs of these mice. This is evidence of metastatic potential. But when you inactivate RUNX2, the metastatic potential is severely limited. So RUNX2 can control this switch to metastasis, and we believe that it does that both in the primary tumor and sustains that in the cells that are becoming metastases. From a functional point of view, ongoing work includes using CRISPR in the model directly to activate and inactivate RUNX2 to explore the consequences of metastases. These are long-term experiments that are ongoing. And we're also using organoid versions of those experiments where we can grow tumors ex vivo, or organoids ex vivo, manipulate genes ex vivo, and then return them to the animal to explore the consequences in vivo. And finally, like I said before, with respect to the gene expression data, the single cell gene expression data, if we look at chromatin signatures and correlate those chromatin signatures, which have been transcribed towards gene expression data, and analyze TCGA data from human lung adenocarcinoma, those tumors that show a pattern associated with our early stage lesions, like module 11, the AT2-like signature, those patients show better prognosis, whereas tumors that show a program that's consistent with our module 11 have worse prognosis compared to those that have a low level of that signature. So once again, there's an association between the programs that are being used in the mouse model and those that are being used in human lung adenocarcinoma. Okay, I've taken a little longer than I wanted to going through those data. I'm happy to have done so, but it means I'm going to have to go quite quickly through the last portion of this talk in order to save time for questions. As I mentioned, we do a lot of work at the level of immune tumor interactions as well. We've done that in the lung quite extensively, and you'll see some references below that describe our analysis of T-cell lung cancer interactions. In order to study the development of immune responses against cancer in these models, it's actually important to provide antigens for them to see. You can observe responses in the absence of known antigens, but the picture becomes much more interesting if you program the tumors with their own antigens. Remember that the tumors don't acquire very many mutations. And so many years ago, Michael DuPage developed a system to allow us to express specific antigens in the developing tumor, thus giving the immune system something to see, and also something for us to track in terms of the specific immune response against those antigens. Here, for example, we use ovalbumin and the synfecal peptide, for which there are tetramers that allow us to stain T cells that that react specifically to that peptide in the context of MHC class 1 to quantify the immune response. I'm not going to review any of the lung data today because much of it that describes the development of these systems has been published. We continue to study it, of course. But I want to turn my attention to pancreas cancer. This is recent work from my group, from Will Friedpaster and Lawrence Lambert, where they want to study the immune response in pancreas cancer, a very deadly cancer, as you likely know, for which survival rates, at 5-year survival rates, are still less than 10%. Immune-based therapies for pancreas cancer have not worked well for the majority of patients. Single and combination immunotherapy really doesn't work very well, except in patients who have mismatch repair mutations where their mutational burden is higher. There is recent work from Bob Vonderheide and Mark O'Hara that show that certain combinations might be revealing better responses, and these combinations include CD40 agonists, perhaps to improve priming in the context of response to pancreas cancer. These are published abstracts, and we're all very interested to see when the papers get published how the studies are reading out. But you'll see that we're going to use CD40 in our preclinical studies as well. So Lawrence and Will have developed 2 different models to create immunogenic pancreas cancer. One involves analogous to our lung studies, viral-induced cancer where the virus expresses a known antigen. They had to modify a method that Monty Winslow's lab had used to initiate pancreas cancer by viral installation into the pancreatic ducts. This works quite well. If you use a Cree virus, you get Hannan lesions, PDAC lesions, and liver metastases with a time frame that's similar to models in which Cree is expressed through transgenic reporters. If you, instead of using Cree, you use a modified virus that expresses a fluorescent protein, here M scarlet, tied to that same synfecal peptide from ovalbumin, you can explore the consequences to the animal of an immune response. And indeed, we can score an immune response when we inject this virus, and you can measure that using that tetramer stain that I mentioned. Here, looking in the peripheral blood 2 weeks after injection, you can see that there are tetramer positive cells in these animals, and you can actually track those cells over time, as shown here in multiple animals. The peak of the response is quite early, and then it's reduced, but in many cases sustained over time. You can also see synfecal positive cells within the tissue of these animals. So the immune response is not just peripheral, it's also local within the tissue. What is the clinical response? Well, interestingly, in the case of this model, we see 3 kinds of responses. Initially, the immune system wins. There is examples here of immunoedited tumors. This is a tumor in the pancreas of an animal, but the tumor is not fluorescent because the immune system is forced selection of cells that no longer express the antigen. In other cases, it's a perfectly normal pancreas where there's no evidence of disease. And we call this immune cleared. And we know that this is immune-based because if we get rid of the CD8 compartment of the immune response, now instead we see fluorescent tumors in 100% of animals. Occasionally, we observe escape in this model, and that looks like this, where there's a tumor and it's maintained fluorescence. So this is a tumor that's still antigen expressing. Here, the tumor has won. The immune system has failed to clear the tumor. The problem is that in this model, the percentage of tumors that show this phenotype as opposed to this or this phenotype is very low. So for subsequent studies, it's quite challenging for us to use this model to explore the nature of the immune response or to use it for preclinical studies. So for that reason, Will and Lawrence actually moved to a different model of an immunogenic pancreas cancer. And here they used organoids, ex vivo grown organoids of different genotypes. This is following Dave Tuvison and Hans Clevers' work, transformed those organoids ex vivo. Those transformed organoids can then be transplanted into the parenchyma of the pancreas, and they will grow into tumors that look exactly like the tumors I was just talking about. exactly like the so-called KPC tumors and exactly like human pancreas cancers. In order to make that model immunogenic, they modified the organoids to express, here again, M. scarlet in a safe harbor locus by knocking the M. scarlet plus synfecal allele into the HIP11 locus homozygously, making it more challenging for the tumors to inactivate the expression or downregulate the expression of this antigen gene. If they introduce those antigen expressing organoids into immunodeficient animals, they will form tumors, and those tumors will glow brightly because there's no immunopressure to lose antigen expression. You can see in an immunocompetent animal that there is a strong immune response against those organoids, against the antigen. It peaks and then declines. And interestingly, in this case, the patterns that we observe clinically fall into three groups. In some instances, the immune system wins. About half the time, we observe normal pancreases with no evidence of disease. Those organoids, transformed organoids, were simply eliminated by the immune system. A small percentage of the time, we see this intermediate phenotype where it's a battle of wills. The immune system is applying pressure, but the tumor is still surviving, and you can see some evidence of antigen-expressing tumor cells. And satisfyingly, about half the time, we observe tumors that grow, and those tumors maintain antigen expression. And this is exactly what we need in order to study this further. We call these progressor tumors. They are continuing to develop despite the existence of an immune response, despite the existence of prolonged antigen expression. We know that there is prolonged antigen expression because we can actually take these tumors out, grow them as organoids, and in this ex vivo context, expose them to CD8 T cells that recognize that antigen in the class 1 context. If you grow these organoids in the absence of those CD8 T cells, they will grow into healthy, plump, fluorescent organoids. But if you add CD8 T cells that can recognize those antigens, that antigen, I should say, then the T cells eliminate those organoids and cancer cells fully. So the tumors grow, continuing to express the antigen. In the right context, the immune system can win. But in the in vivo context, as they are developing, the local microenvironment, immune response is insufficient to clear the tumor. And this is exactly what we'd like to understand better. So using T cell profiling, T cells isolated from these progressor tumors, we've carried out single cell RNA sequencing, again with Aviv Regev's lab. Those antigen positive CD8 T cells derived from these tumors can be clustered as shown here. These clusters are interesting. I don't have much time to tell you about them. I'll focus entirely on this exhausted cluster shown here, which is a high fraction of the cells that we observe. And we call these a dysfunctional or exhausted cluster because they express markers associated with exhaustion and dysfunction. Based on that pattern, we've also isolated T cells from human pancreas cancer patients in collaboration with Omer Yilmaz and George Eng. And these two show patterns quite similar to what we observed in the mouse model, where we find PD-1 positive and importantly TIGIT positive T cells at high frequency. Our attention was called to TIGIT specifically because as we looked at human pancreas cancer specimens, we found that the ligand for TIGIT was expressed in a fair fraction of human pancreas cancers, that ligand called CD155. Whereas PD-L1, a ligand for PD-1, is not well expressed in pancreas cancer. And based on the fact that TIGIT was highly expressed in RT cells, it was highly expressed in human T cells, and the ligand was expressed at high frequency in human pancreas cancers, we decided to do TIGIT-based preclinical testing in this model. We tested in a six-arm study in animals that had immunogenic pancreas cancers growing, either control therapy, PD-1 therapy, TIGIT therapy, a combination of TIGIT and CD40, that priming agonist, PD-1 plus CD40, or the combination of PD-1, TIGIT, and CD40, to see whether we could see biological effect. These treatments were well tolerated in animals. And importantly, and finally, we observed that there were no responses in single agent PD-1 or TIGIT. Combining CD40 with PD-1 or TIGIT had minimal responses, but the triple combination of TIGIT, PD-1, and CD40 showed very strong responses, with a few of the animals showing very durable responses. And based on these data, we're now in discussion with clinical colleagues and drug companies to try to advance this combination therapy, including TIGIT, PD-1, and CD40 in pancreas cancer patients. And so with that, and I apologize for going right up into the hour, I'm going to stop and thank the people that have done the work. I think I've thanked everybody as I've gone through, but I appreciate your time, and I'm happy to stay on the line for a little longer if people want to ask questions. Thanks very much.
Thank you, Tyler. That was just spectacular. You still have well over 900 people online, and you got scores of questions that we'll forward to you so you can take a look and see if there are any you want to address individually to the requester. I'll just ask a couple that try to summarize some of the questions. The first is, you're talking about epigenetic alterations, and you mentioned that targeted resequencing was performed, but to what extent are you confident that some of these alterations in cell state aren't due to genetic lesions in the non-coding genome?
It's a fair point. We have not looked at the whole genome, and therefore it's possible that some of them are a function of mutations in the non-coding genome. So I can't rule it out. I think some of the transitions that we see and the sort of plasticity that we see argues against genetic explanations, but no, we can't rule it out fully.
So then the next question, you mentioned that there was the possibility that the co-clustering of some of the primary cells in the first part of your talk with the metastatic cells could be due to re-trafficking of metastatic cells back into the primary tumors, but for a variety of reasons, you said you didn't think that was the case. Do you mind expounding on some of those reasons?
Yeah, so I think that there's a couple of them. First relates to basically our understanding of how those cells arise through the epigenetic transitions that we've characterized. So we can actually see this progressive development of this state that we call the premetastatic state. And that has been correlated with gene expression and immunostaining that we've done in the same lesion. So we have a pretty good sense of how things move from one step to another, and that's all consistent with the picture that I've painted. In addition, the very location of the lesions that we observe that have this pattern when we find it by, for example, immunofluorescence is more consistent with this naturally arising population within a developing tumor than it is with a cell that's arrived from a distant site.
Great. So I guess the last question is about the cluster 5 cells. Two things. One is human tumors that have that signature enriched. You said they have a poor prognosis. Are they also enriched for any mutations in the human tumors? And secondly, when you look at the cluster 5 cells based on immunohistochemical markers, do they have any particular spatial localization within the tumors?
Yeah, so I can't actually answer the first. I don't know. I suspect there has been an analysis of looking for correlations with particular mutational signatures and the existence of the cluster 5 cells. My sense is that it's not restricted to a particular genotype, but I actually don't know the details. That was analysis that was done in Tuomas's lab specifically. In terms of the spatial localization, I think that's a very important general point and represents, I think, part of what we're trying to achieve in the current and future extension of this work, applying spatial transcriptomics or other spatial methods to figure out where these cells are. We see them present in a more scattered fashion than in a clustered fashion. I showed you some of that with the SLC4A11 staining. So I don't think they represent just small outgrowths of cells locally, but exactly what their neighbors are and what the phenotype of those cells is, we don't have a good sense of. And I think that will require some of the spatial methods that are now being developed and starting to work quite well.
Well, thank you again for just a spectacular lecture. Again, we'll pause. We won't have a lecture tomorrow because of an internal Dana Farber meeting, but we will start back up on Thursday with Dr. Jacks's colleague, Angelica Emmon from the Koch Institute, and then the next day, Dr. David Pellman from Dana Farber. Thank you all again for joining us, and we look forward to seeing you on Thursday.
Thank you.